Yes. Okay. Oh, now it's going a bit too fast. Okay. So the first thing is this paper where they interviewed 1,924 people who replied. I mean, they asked a lot more people, but also the number of people who replied, scientists, about the reproducibility of biomedical research. And what they found here is that the key findings include that 72% of participants agreed that there was a reproducibility crisis. So that's quite a large majority, with 27% saying that the crisis was significant.
Sven, technical, you seem to be a bit far from your mic, or maybe, at least for me, maybe you can go closer.
I'm sitting pretty close to my screen, so I don't know.
Okay, well, okay, just don't speak loud then.
Is it working?
Yeah, it's
Yes.
okay, it's okay. Yeah.
Okay. Wait, where was I? So then they asked also what the scientists thought was the most important cause of this irreproducibility, and 62% of people said the pressure to publish was this. About half of the participants, so 54% had run a replication of their own previous published research, while a little bit more, 57%, had run a replication of another research study. Only 16% of participants indicated that their institution had established procedures to enhance the reproducibility of biomedical research. And 67% felt that their institution valued new research over replication studies. So quite a large number of them found that their institution was not so interested in replication. And so participants also reported few opportunities to obtain funding to attempt or to reproduce a study, with 83% perceived it will be harder to do so than to get funding for a novel study. Oh, yes. Yeah, then here there are two papers on AI in biomedical research, so you can have a look at those if you are interested. The AlphaFold 3 algorithm is now open source, so it can be used by everyone and can be understood what is happening. There is this paper by Stephen Orstat about the retirement age, but I don't have access to the full text, so I only see this short introduction. I don't know what his points are in the article, sadly. And then I was in the media, but I sent that to everyone already. And I think if Walter agrees, we can use the study that I discussed here as a topic for the next journal club. So it's on this, cell type specific proteomic aging clocks, which are quite interesting. There was also the EHA conference, which was both virtual and in person. And here is a picture of the in-person group. And I guess the G is having quite a lot of comments later on about this, so I'm not going to go on. And there is also the upcoming Global Health Span Summit in Saudi Arabia next year. Okay, here this is like a resource rather than a study where they took over 20 million cells from 623 different mouse tissues of various life stages, sexes, genotypes, and from all of that they developed an atlas. So that's a resource you can go and consult. In this study, they found in people from 28 countries, over 90 years of age, so very old people, not 70-year-olds or so, but in really old people. The mean age here was actually was 91. They found that stronger muscle mass was actually associated with lower mortality. And this was true in males and in females. And it was also true when you excluded the people that died 2 years after the start of the study. So the people who were already very ill to begin with. So even then, you still had a significant correlation between lower mortality at stronger muscle mass. So here they look at resilience. So resilience is the ability of the body to withstand a temporal stressor type of event. And they looked at 4 of these events: recovery from anesthesia, restoration of the hemoglobin levels in the blood after a blood draw, the speed of wound healing, and the survival after pathogen exposure in mice. And they looked in mice that were treated with 17-alpha estradiol. That's a compound that extends lifespan in males, but not in females. And they also looked at calorie restriction, which improves lifespan in both males and females. And they found that recovery from anesthesia, blood draw, and pathogen exposure had significant and as expected effects depending on the age. However, wound healing was not affected by aging. But they also found that these measures were actually worsened by the lifespan extending interventions rather than improved, which is what you will expect to happen.
So, sorry, Sven, can you repeat what is worst?
So that these resilience markers, so anesthesia recovery, restoration of hemoglobin levels after blood draw, speed of wound healing, and survival of pathogen exposure were worse after these lifespan extending interventions.
Okay, thank you.
So here they looked at social stress in mice, and they used 2 different types. They used restraint stress, and they used subordination stress. So when you have a more higher ranked male in the group, and restraint stress induced the accumulation of P21 dependent senescent cells, but not P16. While this subordination stress led to P16, but not to P21 dependent senescent cells. So the type of senescent cells is dependent on the type of stressor here. And here they found previous research had shown that if you remove P16 senescent cells, that wound healing is slowed. But when you remove P21 senescent cells, wound healing is actually increased. So again, there are different types of senescent cells, and not all of them are equal in how they behave in all settings. So here there was this open competition for biomarkers of aging. Actually, it was methylation aging. They had this methylation data set of 500 people from 18 to 99 years of age. And the participants were asked to develop novel models to predict chronological age, mortality, and multimorbidity based on this. And many people participated, and they came up with a lot of new innovative ways of developing these methylation clocks that were better than the current existing ones. Yeah, so here this paper is looking at developing epigenetic clocks at cell type specific resolution. And the reason for that is that if you look at, for example, here the blood and the brain, 39% or 12% of the epigenetics clock's accuracy is actually driven by shifts in the number of specific cells. So specific blood cells or specific brain cells. So when you age, there are differences in the amount of various blood cells. And most of these epigenetic clocks just take all blood cells and calculate an age from that. But that means that changes in the abundance of different cells can have a huge impact on the clock. And that's why here they try to look at specific cells and develop such a clock. And that's what they do here. And then here it's pretty much the same, where they use long read sequencing and show that they can take a bulk sample and use through long read sequencing, they can extrapolate from that specific cell type data. And here they use that to develop a microglia, which is the immune cells in the brain, a specific type of clock. I'm not going to go into further details on that. That I'm going to skip. That's for the really interested people in epigenetic clocks. Here, the abstract is relatively obscure in what exactly they are doing, but what they say is that they have applied the Waddington landscape, which is this idea that it's quite old, and it is the idea that you have a pluripotent stem cell, and there is this like valley where the pluripotent stem cell is at the top, and as it differentiates, it goes down on this valley to ever further develop the specialized differentiation. And that is driven, of course, by epigenetic changes. And here they apply this to the information theory of aging, but without reading the full paper, I cannot really say much about exactly what they are doing because it's, the abstract is not very specific. Yeah, so here they looked at 5,000 samples from over 2,000 individuals to look at adult methylation clocks sites, and they looked at in these people from birth to early adulthood to explore what factors may drive the early life factors may drive changes in the epigenetics. And so they find that indeed things that happen at very early age already influence these methylation clocks. So for example, perinatal environmental exposures such as smoking and diet and maternal health can already impact these epigenetic clocks. And here they looked at males versus female, specifically at the X chromosome methylation. And in females, there was not so much age-related changes, but in males, there was more age-related changes. So it means that, having two X chromosomes seems to dampen is the word I was looking for, dampen the age-related epigenetic drift compared to males, which only have a single X chromosome. This is another resource type of paper where they looked at circular RNAs. It's called the circ-H database. So these are circular pieces of RNA, and they have all types of effects, including that they bind microRNAs, for example. And so here they looked at almost 700 samples from 7 species in 21 tissue types that were already in the literature. And they also generated 47 new samples from mice and monkeys. And they added that to that database to obtain over 400,000 unique circular RNAs that are in that database. So it's a database, it's a resource you can further explore. So here you can cross mice, so generating diversity. So rather than what we typically have, which is inbred mice, which are genetic clones, here you try to generate genetic variation in the mice. And when they did that, and they looked at 2,444 mice that were genetically different, and they identified 6 areas in the genome that significantly contributed to the lifespan, that were also independent of the diet or the drug treatment that was being done to these. And they found that they influenced the lifespan in a sex-dependent manner. And there was also one that was diet-specific effect. And they say that together, these 6 new locations, so these 6 new genes, explain half of the estimated heritable variation of lifespan in these studies. So that's another quite nice finding. And then here in humans, for the very first time, variations in the IGF-1 gene are linked to longevity. So they looked at Ashkenazi Jewish centenarians and their offspring, and they identified these 2 new mutations in the IGF-1 gene, and one of them lowers the amount of IGF-1 in your blood, and the other makes that the IGF-1 doesn't bind so well to the receptor anymore. So both will result in less IGF-1 signaling, which we already know in mice is linked to longer lifespan, but now it's actually linked to increases in human lifespan. So that's very nice. Here there is an updated genome sequence of the naked mole rat, where they have actually tried to generate a telomere to telomere assembly, so a complete map of the naked mole rat gene, and they find a whole bunch of new genes that are linked to various phenotypes. Similarly, here they have generated genome maps and also cell lines of various bat species of this group of bats, the Myotis bats, 8 different ones, 8 different species. And they find, for example, that there is quite some variation in, for example, cancer risk among these bats. And so they identify potential genes that explain this. They also find genes that could explain the unique immunity of these bats to DNA viruses. And then here, very similar again, but now the bowhead whale, sorry. Here they also did a new genome sequence and looked. And what they here find is that we know that the bowhead whale is very immune to cancer, despite having a lot of cells, which is, you know, known as the Petos paradox, which probably most of you have heard. And in elephants, for example, you have the same thing, but elephants have more copies of the p53 gene. But bowhead whales have actually another strategy. And one very interesting observation they here make is that when you try to induce cancer in naked mole rat cells, you actually require fewer mutations, fewer ones than in humans. So the bowhead whales should be more susceptible to cancer. You need to make fewer changes. However, what they find is what explains the immunity of the bowhead whale to cancer is that they have very, very good DNA repair compared to humans or other species. And so they have a very unique CRBP protein that is very high levels of this, and this causes very high fidelity of DNA repair. And then here we look at dogs. So we go to all the species in this month's aging news. And here they identify that line 1, which is one group of transposable elements, which are these jumping genes that they are sometimes called, right? And jumping genes cause genome instability. And large dog species, which we know age faster, have a higher loss of methylation at these line 1 sites, and hypomethylation means that they are no longer repressed, so they become more active. So in larger dog breeds that age faster, you actually have more activation of these transposable elements, and so of this genome instability compared to smaller dog species. And here they looked at 20 phenotypes. Again, in dogs, we are looking. So next to body size, they also looked at things like snout ratio, shedding, and things like that. And interestingly enough, they identify drooling as a phenotype linked to aging. So if you drool as a person, that might be relevant. But good. The funny things aside, they identify new potential genes that are linked to longevity in these dogs, and that's the interesting part, of course. And here again, in dogs, they do castration, and they find that actually castration in dogs may be protective against aging. So they looked at various molecular markers, not aging and not lifespan, I mean. So, and they only looked up to 18 weeks post-surgery. So that's pretty, recent, small, short for dogs. So I will be careful, but they did find, for example, that MDA, so that's a type of oxidative damage to lipids, that was lower in castrated animals compared to the controls. That telomere length was also longer in the castrated ones. That mitochondrial DNA copy number was higher, and so on. So here they identify a new species of jellyfish that is able to reverse its development, so from an adult back to a juvenile, which was something that had previously also been seen in this species. So here they develop a new AI platform, H-Extend, and this was used to screen 1.1 billion compounds to identify a lot of potential geroprotective drugs. Here they find that in the mammalian ovary, there are lots of macromolecules that have very long lifespans. So that means they have time to accumulate damage. And of course, that may be one of the reasons why ovaries are so susceptible to aging compared to other organs, because there is less turnover. And here they do multi-omics on human ovary. Again, this is more of a database paper where they collect a lot of data, so I'm not going to go into details here. Here they find in mice that if you reduce the intake of all amino acids by 67%, or specifically reduce the intake of isoleucine, both in male and female mice, starting at 20 months, so in older mice, that metabolic health is improved without reducing calorie intake, and that they also improve aspects of frailty and slow multiple in molecular indicators of aging. However, it should be pointed out that the lower intake of isoleucine reduces grip strength in both sexes, and it has a mixed sexual dimorphic effects on the heart. Exactly what I mean here is not further specified in the abstract. And from the abstract, it seems that lowering all amino acids does not have these negative side effects. So this could be a potentially interesting way to promote healthy aging in humans, if, of course, this applies to humans. This is a phase 2 clinical trial of an immunotherapy against tau, so one of the proteins involved in Alzheimer's disease. And they found it was very safe and well tolerated. They didn't find any immediate effects on the cognitive markers, but of course it's a phase 2, so it's rather small. It's also 24 months, so it's maybe a bit shorter. It's typically, I haven't looked into the details here, but typically phase 2 trials are not powered to detect improvements in the outcome. They are mainly made to look for safety of the drug. But they did find some improvements in loss of specific brain volume of the cortical tissue, but not the overall brain. So yeah, this could be potentially still interested for Alzheimer's disease, but of course a phase 3 trial is necessary to see if there is actually clinical benefit. And here, some people may find this interesting, is that cannabis smoking or oral taking in old mice improved some aspects of memory, but it had no effects on the memory in young mice. So it only works in old mice. Yeah, this is a specific clock for immune aging. Here they show that the loss of the intestinal barrier integrity. So when you get old, your intestinal barrier becomes leaky, and bacterial products like LPS can get in, or even full bacteria could potentially get into the bloodstream. And I link that here to actually aging of the thymus gland, so that if you have higher level of intestinal barrier loss, that you also have higher levels of thymic loss. And then that, of course, leads to T-cell aging. I'm going to skip that. Yeah, this is another atlas, another resource paper. So here, interestingly enough, when you bleed mice multiple times, when you bleed the mice, of course, their hematopoietic stem cells need to get activated to repopulate, to make new blood cells that have been lost by bleeding. And interestingly enough, that actually reduces the number of double-strand breaks, which is DNA damage in these blood cells, in these hematopoietic blood stem cells. So stimulating their proliferation, which you might think is not a good thing, actually seems to protect them against DNA damage. And so here they looked also at these hematopoietic stem cells, and they found that there are two different populations, a CD150 low and high population. And this CD150 low population in old animals, if you transplant those, they actually improve the aging. But if you transplant all the hematopoietic stem cells, or the CD150 high specific ones, then you don't see those benefits. So these CD150 low hematopoietic stem cells are a population, a subpopulation of hematopoietic stem cells in old animals that are actually younger and healthier. And then here from the Church lab, there is a paper where they identified this ATF3 as a cause of skin aging. And when they used mRNA treatments to produce more of this ATF3, because the ATF3 is normally reduced with aging by 20%. And when they increased it, they found, for example, that cell senescence was decreased by 25%, 20% increase in the proliferation of the stem cells. They found increase in collagen production by 200%. So they found that skin aging was slowed and skin health was improved. And that's the scientific news here. I'm going to skip through the C. elegans one. This paper, but I already sent it in the mail with highlights. So that's an interesting one to look at because they asked a whole bunch of aging experts their opinion. Yeah, this one is interesting. And I think that's pretty much it. There are a bunch of reviews you can look at yourself if you are interested. And this is something I wanted to include, but I'm not going to discuss. But for people who want to use, these are like new tools you can use for research. So I'm not going to go into it. So that's it. Questions?